Hey, my name is Andrew Richards. I'm a grad student at UC Davis, and today I'm going to talk about Pierce's disease and how to manage it using biological control agents. First, we'll go over a little bit of background. Pierce's disease is caused by a bacteria called Xylella fastidiosa, and it affects grapevines in California and around the world. This bacteria is vector-borne, and it is transmitted by native as well as introduced sharpshooters. And these sharpshooters require riparian areas to reproduce. And so if you've got a vineyard next to a riparian area, that's going to be at an increased risk of getting Pierce's disease. So when the sharpshooter feeds on the grapevine, the bacteria is introduced into the xylem tissue, and the following symptoms occur. On the left, you have a typical leaf scorching. In the middle picture, you've got shriveling of berries, and on the right, you have altered tissue lignification. So you'll have these green spots surrounded by lignified tissue. To manage Pierce's disease, we established a field trial at UC Davis and artificially inoculated grapevines with Xylella fastidiosa, as well as applied 3 bacterial strains and a phage to combat the disease. So in May of 2023, we applied the 3 different bacteria as well as the phage by themselves in combination. The next day on May 18th, we applied Xylella fastidiosa via a drop puncture method. And then 1 week after, we reapplied all those biological controls. To determine disease progression and disease severity, we counted the percentage of scorched leaves compared to the total number of leaves per shoot. So we did 4 disease ratings in August, 3 in August and 1 in September, where the percentage of scorched leaves of each of the treatments was compared. When the experiment was done, we also collected petioles and extracted DNA and then performed qPCR to determine the concentration of Xylella fastidiosa at the end of the experiment. The results shown here are from the final sampling time. In the figure on the left, you can see percentage of scorched leaves for each biological control treatment. All treatments resulted in a lower percentage of scorched leaves compared to the positive control. And in the figure on the right, you can see qPCR data from DNA extracted from petioles at the end of the experiment. The axis is flipped because the higher the CQ value, the lower concentration of Xylella fastidiosa was found in the sample. And you can see here the qPCR data accompanies the physiological data very well. So all of the applied biological control treatments resulted in a higher CQ value. And again, that means a lower concentration of Xylella fastidiosa in the sample. In conclusion, these results show efficacy for all biological controls to manage Pierce's disease. These results also align with the results from last year's trial, and we will be repeating this trial for this growing season at 2 different locations in Armstrong. I hope you enjoyed the presentation, and thank you very much for listening.